Good morning, everybody. You look as if you had a good sleep. I'm glad for that. Before I hand over to my colleague and friend, Professor Raz Jelinek, I want to welcome everybody again. We have been joined by some of our incredible students from our Stable Care campus. So I encourage you to get to know them as well, and look forward to a great morning. As Danny, you had here? As Danny mentioned at the beginning of the week, or one point during the week, we have some really incredible royal families who have been attached to BGU for many, many years, and one of them is the Kreitman family, who you all know well as a name. And we don't have members of the Kreitman family with us today, but hopefully we will welcome the extended family at the near time in the future. But this is the annual Irene Hyman Kreitman event, and I welcome you, and Professor Jelinek, thank you.
Okay, good morning, everybody. You're on for an exciting session, especially fitting after breakfast, because this session is about food. So after this session, you would know if what you ate for breakfast is good for you or maybe not good for you. So we'll learn it in the next hour or so. First, let me say a few words about the Kreitman family, which this event is after them. This event is held today in honor of the late Irene and Hyman Kreitman, who were among the most loyal and dedicated friends of the university from its inception. Dear Irene and Hyman passionately believed in the importance of creating an institution of higher education in the Negev. They recruited many of their friends around the world to join them in empowering what was then a fledgling institution, and Hyman served as chairman of BGU's Board of Governors. So, through their exceptional generosity, they supported a plethora of projects, including classroom and administration buildings, the award-winning Kreitman Plaza at the Marcus campus, multiple academic chairs, and the internationally recognized Kreitman School of Advanced Graduate Studies, which actually support many of the students who are now sitting in the audience here. So, without further ado, let's start with the session. This session actually highlights food. And this is one, not enough? Okay, so you're gonna have some more food here. Teaser. We are very happy to have received a nice donation from the Mania Eigel Foundation in the UK to launch, to initiate this activity, or activities actually focused on food in all its aspects: food tech, ag tech, nutrition in general, and understanding the molecular basis of nutrition. You're going to hear some of that today, and hopefully next year, when you're going to be here in the next Board of Governors meeting, you're going to hear even more on our achievements and accomplishments in this field. So let me first introduce Professor Iris Shai. Iris is one of our most distinguished nutrition scientists. She's also an adjunct professor at Harvard University. She's been doing groundbreaking research on nutrition, particularly Middle Eastern or Mediterranean nutrition. And I believe we're going to hear about it more now. Please, Iris.
Thank you very much, Raz. Good morning to you. I'm very happy to be here. My pleasure. Okay, so let's start from the good news in this morning. What are the good news? Within 200 years, the life expectancy has more than doubled. So just imagine that 200 years ago, we have been just above 35. That was the life expectancy. Today, the average is a bit below 80, and it's just rising and rising. Good to know. But you know, we're in Israel. We have even less good news. What are the less good news? This is not the same as we were programmed. Actually, physiological and mental functioning, our best time is between the age of 70 to 45, 50. After that, total loss. But this is, no, we have some hope. No, but we're here, right? Because we do plan to live much more, right? And now this is the big question. How do we keep or just attenuate it, this aging? And more than that, even from one side, we do know that life expectancy is rising and rising. From the other sides, it's not the same for the children of today, which are so, so ill, and their life expectancy could be halted. So one of the best ways, or one of the best to actually keep us healthy is, of course, nutrition. And we have a lot of myths about nutrition. We are overwhelmed about the stories, what is good and what is bad. Is it coffee good for you or bad for you? Olive oil, too oily, is it good for you or bad? Macrobiome, yogurt, brain, wine, okay. We have more of the inconsistent results about that, and we do need, but actually you know what is good for you. We are, the scientists are here to give the evidence-based nutrition about that. And why we are so conflicted about that? Because most of the data that you receive are coming out from observational study, anecdotal studies. But the higher way, actually, to give the proof of concept, and the proof is randomized controlled trials. This is the way how drugs are being conducted, intervention trials. And this is what we are doing. So good morning, my name is Shai. This is what we are doing in Israel, in Ben-Gurion University. We conduct long-term, large-scale dietary randomized controlled trials, which are a quite unique niche in the world. I'm also the chair of the Health and Nutrition Innovative International Research Center, and I would like to ask you, or maybe to a bit give a glimpse about do diets work? So this is what we have conducted in the last 15 years. 4 big trials. In the first one, we just ask ourselves, do we have, and in those trials, we take hundreds of participants over about 2 years. We try to use cutting-edge technologies. We have international network, and we ask very simple questions. In the first questions, we try to address new paradigms in dietary strategies. In the second question, we try to see, is it true that moderate alcohol is effective and safe for diabetics? In the third one, we already used MRI to see whether there are distinct methods, interventions, to treat the mobilization of specific fat storage inside our body, to change our body composition. And recently, we completed the next trial. We tried to see what are the effects of amplified dietary polyphenols, which we will discuss about them a lot today, about the gut-fat-brain axis. Okay. So I took 3 questions, and I will try quickly to help to discuss them. The first is, can diet really affect my age-related cognitive decline? Okay, so this is a normal aged brain. And when we are growing and aging, not all of us, of course, and of course except us in this room, but 2 main things happen. First of all, we lose the gray matter. For example, the hippocampus, which is here, the volume is actually going down, and this is the main function of memory. And this is how it looks like for someone which weakens his hippocampus and he has dementia. The second thing is that we enlarge our holes, which are empty and getting more with fluids. So this is the main things, to squeeze our hippocampus and to enlarge the holes. Here you see 284 brains of people that we did for them MRI. And you see that just over the age, you see that mainly from the age of 50, the hippocampus is shrinking. And this is very natural. These people are walking, actually, very close to here. Yes, in the high-tech, in the Nuclear Research Center. So these are people who have cognition function, but this is what happens. The good news are that we have here variation. Not all the people are according to the predicted line. Some people do maintain very high hippocampus levels. The same people, you see, that the more they're aging, their lateral ventricular volume are expanded, and specifically from the age of 50. What can we do? We learn that after almost 2 years of intervention, when we put people on exercise, but we change their lifestyle to more Mediterranean pattern, more polyphenols, and I would like to say in one sentence what are polyphenols. You know, there are secondary metabolites of plants that are performed by plants to help themselves, to protect themselves from pathogens, UVs, stressors of the plants. And you know them like olive oil, coffee, dark chocolate, apple, green leaves, walnuts, green tea, etc. And when people consume those polyphenols, they halted the attenuation of the age-related atrophy by 50%. This is something amazing, and that was actually reported in, if you can get into it, in New York Times report 2 weeks ago. Second question, can diet affect my microbiome? Very, very quick, we know what is microbiome. We have a lot of microbes. Your body contains 150 times more of the DNA related to your germs other than that one you inherited from your parents. Do you know that? So who controls your body? Actually, the microbiome, but we could control them. And there is a crosstalk between the health of us and the microbiome, and maybe Raz will elaborate about more. About 2 kilograms of your body, actually, it's your microbiome, it's not yours. So you can take off from the scale. We did something that is kind of a science fiction. And imagine, I will go to the end. Imagine that when you were young, when you were 20 years old, young, beautiful, healthy, yes? And we could preserve your healthy microbiome then. And we could transplant your autologous microbiome back when you are here now. What would happen to you? We asked the question in another design. We took the 300 participants. We had them to improve and optimize their microbiome. We had them to lose weight, to optimize by polyphenols. We performed their own capsules from their own microbiome. That was happened in BGU, the first time ever trial that has been conducted in the world. And we had them to swallow their own microbiome later in their life. And we learned a few things. That we could initiate microbiome memory, that we could preserve specific microbiome, and we know which of them, and those microbiomes specifically could maintain, for example, our glycemic control. And this is very quickly. And the third one, can diet affect my DNA methylation age? Very, very quickly, because Deborah will talk about it. So we learned so far that our chronological age is not the only one. And some of the things that could affect our health is how our DNA is expressed. And did you know that you have switched on the tailors of your DNA on and off of methylation groups, chemical groups that could express in different levels the genes that you have. So it happens, and this methylation age is much more accurate to predict your mortality and morbidity. And we learned that we could improve our genetic age or methylation age. Actually, when we use almost 300 CPGs, like sites of DNAs, in humans, not in animals, and we did intervention trials with healthy diets, we could attenuate the aging by the DNA, and actually we could make the people after the intervention, in terms of DNA, 8 months younger. This is amazing, and our next step is to discuss and address the epigenetic diet. Indeed, we could maybe modify and improve our health and age by the healthy lifestyle. And I will end up with the future of our group. Again, I'm very happy and proud to be in Ben-Gurion University. In a personal matter, I would like to say that this is a young university that helps us to dream and to think about crazy ideas and to perform them. And the future is personalized medicine. Actually, the next thing is to integrate all the data that we have, over thousands of people that we already learned about them so much. We have data that actually no one in the world has, thousands of MRI images, the responses to the interventions over the years. And we are going now to ask them to come back. It's called the FIT Medical Bank. FIT, it's follow intervention trials. And we try to learn after, we are going to, for example, now have again 1,000 whole brain MRIs to those people, 1,000 liver MRIs, 1,000 abdominal fat MRIs. We are going to collect the macrobiome, to collect all the samples that we can collect for humans, to go for omics, for DNA, to create national/BGU data that could help us to predict specific health status, like aging status of those people. This is what we are going to do in the next years. And indeed, your body appreciates your healthy lifestyle. I would like to thank you first for attention. These are our true collaborators all over the world that are in our papers. And you're actually welcome to enjoy your polyphenols. Thank you.
Thank you very much, Iris. So you're welcome to try some polyphenols. They're going to give it out now. Just to mention, some of the money, significant parts of the money from the Mania Eigel donation will actually go towards this large-scale trial that Iris was telling us about. So again, if you come here in 1 year, 2 years, we're going to hopefully hear about the exciting results and how we can increase our lifespan by 10 years, Iris. We are with the business of pledges here. So how much you pledge, how many years extra you pledge that we're going to have with this? For $1 million, we pledge 5 extra years. Any takers? What? Okay, yes, sure. So, living longer, talking about living longer, we're going to move on to the next lecture. The next lecture is going to be given by a chemist who also happens to be myself. And okay, so let's start. Okay. So, we often see these ads. Or this ad. Nothing happens. Or this ad. It doesn't move. Hey. Or this ad. Okay, so we see all these ads. You know, all of them show blue skies. All of them show smiley people sometime. And they all seem to promise us good health only if we open our wallets and we go to the supermarket and we buy these products. We buy these yogurts. But no. Okay. Restarting. Yoav, what's going on? Okay, we're back here. Should I do this or that? Okay, so the question is, do milk fermented probiotics, yogurt, does it truly have health benefits? The question is, Yamina Vesmola. Okay, the answer is actually we don't know. Despite the fact that when we go to the supermarket and we see aisles over aisles of yogurt and other probiotic milk products, and it's multi-billion dollar industries, we actually don't have a solid scientific basis for these claims. We don't know. And the goal of this research that I'm going to tell you about is specifically designed to answer this question. Do probiotic milk, or specifically, does yogurt really help your health? Now, behind every scientific story, there is a person. And behind every person, as we all know, there is a story. And the story of this yogurt has to do with people. I am the minor actor, okay? But I still need to tell you a little bit about myself because it is relevant to the story of the yogurt. So this is me, okay, on good days, after eating yogurt. Let me tell you a little bit about myself, okay? I'm married. I have 4 children. I have been the chemistry department chairman. This is a few years ago, okay? I'm a full professor. Around 10 years ago, I ran in the election to the Faculty of Natural Science Dean. I ended up in the 3rd place out of 3. Why am I showing you this list? Okay, so you see this list, okay? All the checks. This is the best recipe for a midlife crisis. Okay, I did quite a lot, okay? I was approaching 50, okay? And I did all these things, and then I was asking myself, what's next? Okay, like a man in his midlife crisis. And then came this person, Orit. And the story of the yogurt, it's really her story, okay? Now, Orit came and knocked on my door, and she asked me to do a PhD. Now, Orit was not your conventional PhD student or prospective PhD student. She's a mother of 4. When she came to me, she already spent almost 20 years in industry. She worked in Intel. She had quite a senior position in Intel. She worked at Perrigo. Okay, these are major companies. But then she said, Raz, my dream is to do a PhD. I want to do a PhD with you. I said, sure, I'm happy to have students in my lab. But then she said, look, I want to do a special project for a PhD. And then she told me a story. My son had really bad intestinal problems that could not be healed, could not be taken care of by conventional medicine. And she told me, I found this yogurt, this special yogurt actually called Kefir. And this Kefir I gave to my son, and after several weeks, he felt much, much better. And since then, his stomach is okay. And Ori told me, I want to study this Kefir. I want to find out what is the scientific basis of this presumptive therapeutic effect. Now, I told Orit, look, I'm a chemist, a simple chemist. I'm not a food scientist, I'm not a microbiologist. But remember this, what's next? I said, okay, I'm willing to do that. You're going to take the lead, Orit. But because I'm open to anything right now, I'm in a midlife crisis, etc., I'm going to go with you. So this is the story. Now let me tell you a little bit about kefir, okay? Kefir actually, different than yogurt. When you go to the supermarket and you buy yogurt usually has 1 or 2 probiotic bacteria. That's what supposedly give the yogurt its health benefits. Now kefir has many, many more microorganisms, many more probiotic microorganisms. Now think about it. Kefir has these millions and trillions of microorganisms from different species. Each one of them secretes to the milk various molecules. The secret is to find the molecules and to demonstrate what they do biologically. Now, essentially, this is like looking for a needle in a haystack. There's all these microorganisms. There's all these molecules that they secrete. To find 1 or 2 molecules that actually do the magic, it's a major effort. And indeed, it took 7 years, 7 long years of Orit's time to find these 2 molecules. Aren't they beautiful? These are my babies. These are small chemical molecules, okay? You see, you see the hydrogen, the oxygen, we see rings here. These 2 small molecules are the molecules, or some of the molecules, that we found are so special and do incredible things to our health in this kefir. Now, the first thing we studied was obviously the intestinal issues, okay? And in very long experiments that we did with mice, we found that these 2 molecules help chronic inflammation in your intestine. There are very bad diseases that are becoming more and more prevalent now that involve chronic inflammation of the gut. Diseases like Crohn's disease, you might have heard, colitis. All these diseases are characterized by chronic inflammation. What we found was that these 2 molecules that I showed you really attenuates the inflammation in the intestine. And interestingly, even this kefir that we gave to some people that have colitis and Crohn's disease, they reported much better feeling and much better health outcome. It was incredible. So that was the first exciting discovery. And then, March 2020. Do you remember what happened in March 2020? This thing, this unexpected guest, uninvited guest, appeared in our lives. Now, why am I mentioning COVID-19 here? Now, you might know that the main reason people become so ill and sometimes die with COVID-19 is not specifically the virus, but it is the inflammation, the severe inflammation that this virus is causing us. Now, the amazing thing about, the very smart thing from the virus point of view, okay, that the virus does, the virus goes all over our body and induces severe inflammation all over our body, in different organs, in the lungs, in the liver, in the intestine, in the brain. Sometimes this inflammation is so severe, it's also called cytokine storm. You might have heard the expression. This inflammation becomes so severe that the body essentially attacks itself, and then you might die. Now remember the word inflammation here. So we have a viral disease that causes severe inflammation. Then we have this kefir and these two molecules that supposedly, what we found with these intestinal diseases, these molecules have anti-inflammation effects. So we made 1 and 1, and we said, okay, let's test whether these two molecules that we found from the kefir have an effect against this severe inflammation. Now, naturally, we couldn't give these molecules to COVID patients, although we really, really wanted, because we believed in this, but the Ministry of Health naturally did not let us do that. But then we did mice experiments. Okay, so we're gonna see now some experiments with mice. Okay, mice that we did not give them COVID infection. This is something you cannot do for ethical reason. But you can make the mice extremely ill. You can give them severe inflammation. And you're going to see some short videos of these poor or happy mice, whatever you want to call them. Okay, so here you see mice that are sick. They have severe inflammation. So you see, just like people, they are shaking, they have high fever, they don't move. Okay, when you're sick with COVID, some people in the audience can attest, even our president. You lie in bed and you just don't want to go anywhere, okay? But then, now I need your help because you need to press the movie. These are the same mice, okay? No, you need to press on the movie. It's a movie here. Okay, thanks. So you see, these mice on the right were given the two molecules orally, just like in their water. We didn't inject them in the veins or anything. We just gave them to drink these two molecules. And you can see these mice are back to normal. They're healthy. They're playing, okay? Just like us, you know, when we're active, we make tunnels. So what we see here is complete 100% healing of mice with severe inflammation. Now think about it. COVID patients, people that are very sick with COVID, getting these molecules, drinking them, the next day, get up happy. The truth of the matter that I offered Danny these two molecules when he was sick, but probably he was too sick to read my WhatsApp. Now, I'm a chemist actually, so as a chemist, I try to understand the mechanism, okay? So it's not enough to show happy mice versus sad mice. I really want to understand what's the molecular basis. And what you see here is actually our work or our understanding of the molecular mechanism. We see here a very famous protein, an immune protein that nobody knew actually that it gets involved in COVID-related or inflammation-related activities. We found that these two molecules really affect directly this protein, which is chemistry-wise, it's actually a major discovery. Together, actually, we got unexpected help from this person. I'm sure you know him. Beside his, hobby as a president of the university, he's also a biologist. And lucky for me, occasionally I have time to sit with him and talk with him about minor issues as R&D in the university, but sometimes I can talk to him science. And in one of these conversations, we made a connection. Because Danny recognized this story, and he suggested that we look at this protein. Sure enough, that's the target. So we're very lucky that we have a biologist as a president. So, all you need is kefir. It helps COVID. When, I had 3 vaccines and I was sick with COVID. I don't know if it's because of this kefir that I drink every morning, but I had it very, very mild. One afternoon with little fever, next day, great. And knock on the wood, I'm okay. All because of the kefir. And some other things. It helps intestinal problems. It also has antibacterial properties, which I didn't tell you about. And it actually also helps wounds. When you have wounds in your body, on your skin, you basically have inflammation. We find that this yogurt, when you put it on your skin, can really help inflammation. And really, it's only the beginning. Sky's the limit. So, l'chaim, and there are some, we have some yogurt here, if those of you want to live like 2 weeks longer or a year longer. I'm going to drink the first one so you see it's not poisonous. Sure. Yeah. Excellent question. The question was, is there any gender difference with kefir? Now, I don't know about people, but with mice, we find that female mice were more sensitive to inflammation, but they were more, they got healed better and faster. So, yes.
I have been drinking almost a bottle of kefir, first thing in the morning and last thing in the night for the last 5 years.
Can I use you as a presenter here? Fantastic. Yeah.
Is there any difference between cow milk and goat milk?
Another excellent question. The truth of the matter, I don't know. What you're drinking now, this is kefir from goat milk. Beside my hobby as a vice president, I also make my own cheese and own yogurt. I go to a farmer and I buy raw milk. This is not sterilized, pasteurized, so don't worry. But there definitely might be differences between goat and sheep and cow's milk. This is something that... Well, I don't want to talk about the company. What? Where do I get the kefir? Some kefir you can buy in the supermarket, actually, but this kefir, the composition of this kefir is actually different than what you can buy commercially. The individual microorganisms here you can buy, but this composition is quite special. You know, if there are some people in the audience that have spare $2 million, we take investments. Yes. Well, I drink approximately full cup of kefir every morning, and not a bottle. I drink it with granola. So I'm going to open a business. Okay. Now, I'm going to be here for the rest of the day for questions. I apologize, but we are short of time. Got carried away. So thank you. Our next lecture, we switch gears. We had yogurt, we had polyphenols, now we're going to have wine. And unfortunately, the Iron Fate, who is leading this research as Deborah, could not be here. Instead, we have his students. Kidaneamraym Reta arrived, he's a PhD student from Ethiopia. Do they drink wine in Ethiopia?
Yes.
Good. Well, good to know. Good to know. He's going to tell us about wine in the desert. Please.
Okay, thank you. Before a year, one important thing happened to me. I heard one exciting information from one of my friends that's studying here for his PhD. He told me about an opportunity that Professor Aaron Veit and Professor Naftali Lazarovich looking for. At that time, I was very excited. Do you know why? I had very deep interest to study on a dryland plant which is vulnerable to climatic change. Besides that, since the beginning of my childhood, when I heard about Israel, I had a heartfelt happiness, and it was my passion, and it was my wish to be here once a time. And immediately I made a contact with them. Okay. And then I made an interview. Finally, they say yes for my interview. So I realized that my dream comes true. And now I'm here doing my research in Ben-Gurion University, living in my dream country. My name is Kidaneamraym. You can call me Kidane in short. I am from Ethiopia. This green picture that you are looking now is from Ethiopia, which belongs to Awash Winery, one of the biggest and oldest winery in Ethiopia. And this gorgeous green vineyard also from Italy, where Professor Aaron Veit was born. Let's come back to this, from these two green pictures to the heroic viticulture in the Negev desert here in Israel. As we all know, grapevine is one of the largest fruit crops that can produce about 78 million tons per year, especially in the last 5 years, except a little ups and downs because of the pandemic. In many countries, specifically to Middle East and Europe regions, viticulture and winemaking are the key socio-economic industries. Literally, grapevine has been tied to many of agricultural and religious activities since the beginning of time. As an indication of earliest excavation from Jericho, at early Bronze Age, around 3000 BC, is an indication of the cultivation of grapevine has been started since the ancient of time. Contemporary seeds also excavated from the desert. Similarly, seeds from the Iron Age have been found in many excavations in Israel. This is Shifta ruins, one of the 5 ruins that found in the Israel Negev desert. This ruins was during the Nabatean people around 800 to 100 AD. As you can see from these 2 holes, the ancient people at that time have been using for wine pressing. And as you can see from these 2 biblical verses, grapevine, vineyard, and wine, cited 100 times, practically in many books of the Bible. This is Ramat Negev, which comprises 22% of the Israel land, which is mainly characterized by low rainfall with huge amount of water loss through evaporation many mechanisms. This desert uniquely suited to demystifying elemental terrors, perhaps learning how to adjust them for climatic change. And this rocky desert lacks the nutrients and basic necessities for the growth of the grapevine. So growers must reach the soil by adding one element at a time. And this can allow scientists to look at each individual element, their influence on the growth of the plant, and to see the resulting taste of the wine. You may be surprised here. And this is what existing now in the Negev desert, which is surrounded by a rocky hill. This breathtaking view of the vineyard found in between of Beersheba and Mizpeh Ramon, in combination with the ancient wine process and production with the current innovative initiative of the wineries and vineyard, makes the Negev desert a sweet destination for the global tourism. The Negev desert, rich in many wineries and vineyards that can prove the world the possibility of producing quality wine from the harsh condition in the desert. As you can see this, the little beautiful daughter, in fact, this girl is Aaron's daughter, tasting with her fingertip. But also you are going to taste soon. Maybe some of you tasted yesterday in our campus from our tasting wine. And this, we have another big challenge that arose from climatic change. We have a challenge for drought, salinity, high temperature, and seasonal migratory birds. The Negev deserts are rich seasonally with migratory birds, and this migratory bird results to lose about 30% of the vineyard yield. When it comes to other specific entity, which is salinity, it affects the whole vine system, not only the grapevine, but also other croplands that can grow in this area. By 2050, it's projected that 50% of the global arable land will affect by salinity. Here we have another big problem that arose from high temperature. As you can see, this berry is highly affected due to high temperature. During the maturity time, here in the Negev desert, the temperature hits about 50 degrees centigrade for 2 months, day and night. Imagine how it can affect this lovely vineyard. At this time, due to the high temperature, the berries will accumulate high amount of hydrogen peroxide. And this accumulation results to hastened ripening process. And this hastened ripening process will result to shrinking and bursting of the berries. Then finally, the berry will lose its volatile content, its aromatic content, and its flavor, but also the phenol, which is very important for health and for tasting. This is what we are doing now in Ben-Gurion University. As scientific people, we have a challenge, but we need to switch this challenge into an opportunity. We have an opportunity to select best varieties that can grow in this environment by selecting from the biodiversity that we have. And selecting a best variety could not enough to produce the required yield and quality. What we have to add, we need to add management practice, like by enhancing the cluster through netting just to protect from the insects, the attacking bird, and many others. We can enhance the vineyard condition to lower the temperature and to maintain the optimum relative humidity, and many other things. But there is another more thing which is more important that I like to study to know what is happening inside. That is the hidden half of the plant part found underneath. The interaction between the roots with the soil, the water, the soil microbe, and the salinity. In our experimental vineyard established in our campus, we are studying about it by applying differential level of salinity, irrigation on different rootstocks to determine the threshold level of the plant tolerance for drought and salinity. So now the Negev desert attracts the attention from the scientific person, from the government, and from the donor. When it comes to the information dissemination, the Negev desert attracts the eye of many international mainstreaming media, including the New York Times and many other websites. This is another way of communication that is scientific publication. Professor Aaron White has more than 100 scientific papers published in many different research topics. As you can see from this picture, he has around here inside Boker, and he has tremendous network with people that have similar research interest in this area. He has many connections with many of European countries, in the US, and many other countries. This is our state of art in our research group. We have an integrated discipline, like using a drone-based imaging system to detect the thermal stress on the vineyard that could happen. We have another component that is high-throughput analytical tools. And the data that we found from these two components will integrate using a modern statistical tools and modern statistical models. And this can help us to predict our result, what we want to see, a healthy vineyard with a tasty glass from the non-predictable world. To bring this, we are here. For those of who didn't know, Professor Aaron White is this one. Maybe most of you know. we are from most of the world, from most of the continent, and we can say that our lab comprised the little world. And so now we are working on the grapevine to bring a good quality wine for our globe. Thank you very much.
Beautiful, very nice. Beautiful. So we want you to live this session both full with food, healthy food, and also happy with wine. So the Negev wine. So let's continue with one of our young and bright scientists, Dr. Debbie Toiber from the Department of Life Sciences. And she's going to tell us something very insightful about the relationship between DNA and nutrition. Please, Debbie.
Great, thank you. Here's my presentation. Let's see. Okay, almost there. Wait, it's the last talk in the session, so be patient and we can start. So we are constantly bombarded by anti-aging products if we have to take this cream or supplement or all these advertisements that want to keep us young. And why are we so afraid of aging? And with aging come age-related diseases. We have more chances of developing immune system decline, heart disease, metabolic syndrome, and cognitive decline, such as Alzheimer's and Parkinson's disease. So, of course, to try to prevent this, we have to understand why do we age. And one of the main theories in the aging field is that we accumulate DNA damage every day in our every life. We have to use our DNA to copy and to prepare new proteins, and also it is constantly attacked by metabolites and by UV light, the sunlight, chemicals. So it's constantly being damaged. And when it gets damaged, we can repair the damage, and that will be ideal. But of course, biological machines are not perfect. And then we cannot repair all of it. So we are constantly accumulating damage, and as we live longer, we have more chances of this accumulation of DNA damage. What happens with these damaged cells? If we feel that we have too much damage, our cells can be altruistic and commit suicide, decide, I am damaged, I can be bad for the surrounding cells, so maybe I commit suicide and prevent damaging other cells. But of course, if too many of these cells die, we are starting to lose tissue. We lose tissue capacity. Another option is senescence. We have these cells in our body that are like old cells that are constantly sending signals to the other cells, I am still here, but I am not okay, and sending pro-inflammatory signals like, I am not okay, but I'm here, count on me. But really, at the end of the day, they are doing damage also to the tissue. And last, if we have damage and we don't understand that we have this damage and the cells keep dividing, then we get a tumor and we can get cancer. So all of these age-related diseases can come from accumulation of DNA damage. One of the things that is also controlling how our body functions is the epigenetic code. So if we imagine that every cell in our body has the same DNA, right? We have the same DNA in every cell of our body, how these cells are different, and this is through the epigenetic code. So if you imagine that our genetic code is a script for a play where all the actors get this script, but each actor has to say something different because we have to have different functions. So the epigenetic code will be these marks that we put on the script for every actor. In this case, a brain cell has the script that is telling us to produce things that allow us to think, while a muscle cell may be preparing things that help us build strength and movement. So this epigenetic code can also be influenced by the environment. So we have the identity of the cells, but also we can help our epigenetic code to regulate how we should say the things. Like if we are in a play and try some phrase in a certain way, it may work well and everybody was enjoying the play. But maybe if we say it differently, they didn't enjoy. So we can adapt and do some changes on how we use the epigenetic code. And there are things that are good for us and bad for us. Diet, sport, depression, trauma, all these things can affect how we read this epigenetic code. And one of the things that affect the epigenetic code is DNA damage. Imagine that we damage the script, we have to remove everything, repair the script, and then put back all the marks that were there. But with time, we don't manage to put back all these marks, and then we start reading phrases that are not supposed to be there. We start saying phrases that are belonging to someone else, and this may cause disease. So how do we study aging in my lab? And a little bit about me. I came from Mexico. I made Aliyah when I was 19 by myself and studied in Jerusalem in the Professor Sorek's lab, neurodegeneration. Then I moved to Harvard Medical School where I studied epigenetics and aging through DNA repair. And when I opened my lab in Ben-Gurion University, I decided to put these two things together and study aging and neurodegeneration through molecular mechanisms such as DNA repair. So how can we study aging? We take one gene, CIR6 gene. It's not important that you remember this name. It's a model. And this gene is an enzyme that can repair the DNA damage. We discovered that this is a first responder. When there is DNA damage, it arrives really, really fast to the sites of damage and starts sending signals to everyone what to do. Come, repair, remove this epigenetic code, and then we can repair our damage. The second thing that it does, it's modify the epigenetic code. It adds these marks on the script to tell, read me, don't read me. So when we don't have it, we have a problem. If we remove in our animal models these genes, they have progeria. They have an accelerated aging. They look like these really old kids that age really fast. So we have this in our mice. If you don't have this repair capacity, you age very fast. On the other side, if we add extra copies, the mice can have an extended lifespan. So we know this is a very important factor in aging and how do we age. We know also, to our bad luck, that with age we have increased risk of developing a neurodegenerative disease. There are only about 5% of the cases of Alzheimer's disease that are genetic, but most of them are sporadic. We don't really know the cause, but we know it's age-related. And as we get older, our risk increases. So we want to understand what is happening as we age that is affecting our brain, and if we can do something about it. And we see that CIR6 is really, really decreased in Alzheimer's patients. We have less of this protective gene. Now, we are born with this gene. We have it in our cells, but we are not using it. We close this epigenetic code. The actors are not reading this phrase, and then we are less protected. So we want to understand why it's decreased. And one of the factors that we found is diet. If we feed our animals on a normal diet, they have good levels and they are protected. But if we feed them on a high-fat diet, they have reduced levels of this CIR6 that is supposed to help us prevent neurodegeneration. So we already know from many, many studies that a good, healthy aging can be prevent disease through exercise and all the things that you hear in these talks and good diet. And if we have a metabolic syndrome, obesity, a sedentary life, we increase our chances of developing a neurodegenerative disease. But why? We are molecular biologists. We want to understand why, who are the players. And for this, we use our animal model. So imagine that we have mice that have two copies, one from the mom and one from the dad of this CIR6. So we can repair our epigenetic code and our DNA. We have fed them in a normal diet, only one copy. One copy is more similar to what happens with aging. This level of this protein declines or is in Alzheimer's disease. Or 0 copies, that they are really impaired and have neurodegeneration from very young age. And we measure how they learn, if these mice can learn and remember things. And of course, 2 copies, they are all doing very well. 1 copy on a normal diet, they are doing really well. But 0 copies, they don't learn, they have neurodegeneration. If we challenge these animals on a high-fat diet, we see that the 2 copies have a slight decline, but still are doing fine. But if we have only 1 copy, on a normal diet, they were doing fine. But on a high-fat diet, they are developing neurodegeneration and the cognitive decline. And 0 copies, of course, they are doing worse. So we want to understand how this is happening because we don't know which genes we got from our parents. We may get good genes, bad genes, we may get good copies that express a lot. But we know that our behavior our interaction with the environment, maybe through diet, can influence the outcome. One copy, but good diet, we won't have any problem. Bad copy, but bad diet, then we'll develop disease. So we also try to understand, okay, what can we do about it? And for this, we analyze the brains of Parkinson's disease, Alzheimer's disease, regular aging through the focusing lens of SIR6. And one intervention that was shown to prevent in animal models several of the characteristics of aging is calorie restriction. Of course, this calorie restriction is not fun. No one really wants to do it. It's very difficult. It's 30% less calories, and we want to understand what is the molecular mechanism. Maybe we can target it. So for this, we found that, of course, there are changes that are happening with aging, normal aging, changes that happen with neurodegeneration, but some of them can be reversed by calorie restriction. So we want to understand what is happening at the molecular level. And what we see, if all these gray things are the genes that are changed with aging and with neurodegenerative diseases, we can see in color which ones are the influencers, because if I need to change 100 genes, it's very difficult. But if I find who is regulating all of them, for example, here in yellow, this one is an influencer for the reversible genes. So we can target this in the future to allow us to have like a calorie restriction effect in our body. So we don't know right now where our brain is heading, right? We don't know if in 20 years we will develop a neurodegenerative disease. But we want to do some things and interventions to prevent this. And this investigation can help us also to know where are we going, where is our brain heading, but also if we are starting to do some intervention, if this is helping, if this is changing the signature that was going towards neurodegeneration or healthy aging. And of course, we can find better diets or molecular and chemical compounds, like you heard from the previous talks, that can help us prevent aging and neurodegeneration, and at the molecular level, predict the efficiency of these treatments and prevent and understand neurodegeneration. So this was done by an amazing group and my amazing students in the lab, and thank you for all the funding. And of course, thank you very much for hanging in here for my talk, and thank you very much. Okay, so we wanted to give you some brain games so you can start playing and keep your brain sharp. I am not giving you food because calorie restriction is good for you, but I don't want to put you in that, so don't worry.
Wonderful. Thank you very much, Debbie. I'd like to thank all the speakers of this session. Go out, eat healthy, and see how we can improve on that. Thank you very much.